The mystery of why some people get COVID-19 much more severely than others, many of whom seem to be mostly unaffected by it. Maybe we have a really important insight into that from a recent paper that came out today. We're going to be discussing T cells, science, why some people get walloped. Maybe we have a good explanation for here's why. Hello, everyone. Dr. Chris Martenson here with your July 16th update on SARS-CoV-2. Let's just jump right in. Not even going to do the numbers today. And we've got a number of papers coming out, like this one right here from the Karolinka Institute right here. All these people, really interesting paper called Robust T Cell Immunity in Convalescent Individuals with Asymptomatic or Mild COVID-19. Super interesting paper. But the one I really want to get to today is this one that just came out in Nature on July 7th. Took me a while to get through it and really look at this. So it was published online just on the 15th of July here. And title, SARS-CoV-2 specific T cell immunity in cases of COVID-19 and SARS and uninfected controls. So they're looking at T cells. And as a reminder, because we've been going through this for a while, here's your immune system. And it has this innate side, which is the rapid response. It has this adaptive immunity, sort of a slow response. And over here on this side, we have these things called T cells. You've got the CD4 plus and CD8 plus T cells, and they've got very, very specific things that they do. One of them is the CD cells can retain long-term memory. They get exposed to some pathogen. Some of them just keep that in their memory, and then they lurk around and they wait until they see that immune response antigen come along again, and then they burst into action. And of course, your immune system, very, very complicated. All these different actors, mast cells, dendritic cells, macrophages, natural killer T cells, basophils, granulocytes, eosinophils, all these things, right? And in the middle, you have these odd T cells here and your natural killer T cells. And then your B cells making the antibodies over here. But the T cells are what we're going to talk about here today. So that's what this title was talking about, SARS-CoV-2 specific T cell immunity in cases of COVID-19 and SARS, and this is the interesting part, uninfected controls. So this is very interesting. It's worth going through. And the reason for this is, well, hey, this is a big medical mystery, and we're trying to figure this whole thing out. So it's a little bit of the Sherlock Holmes in me that says, I'm really curious. I want to understand what's going on. And the second thing, though, is that what really matters about getting back to our lives, such as we can after all of this debt comes down on us like a rainstorm, is, are we going to get herd immunity? Are we going to get vaccines? Are we going to get, how are we going to get to this? This T cell response gives us really important clues to why it is that some people are seemingly resistant, if not completely immune or pre-immune even to COVID, and other people just get really walloped. And once we know that, we'll have a better shot of understanding, is a vaccine, which is really going to be coming down this B cell pathway right here. This is where a vaccine is trying to generate antibodies. But maybe that's, according to this paper, that might not be the place we want to focus our efforts. It may well be that it's the T cell side of things that's really where the action is. So let's go into this. Let's look at it. I've done the level best I can to understand this paper. Not my area of expertise. I'm not an immunologist, never wanted to be one, never even wanted to know this much about it. But now that we're in the middle of COVID, here we are. Let's figure this all out together as best we can. So I'll do what I can. Again, if I get something wrong, just help me out, let me know, and we'll fix it. So first thing, let's go straight here into the findings that they had here in this paper. And let me see if is that exactly where I want to start? Yeah, I guess so. Memory T cells. Whoops, wrong pen. Get the yellow one. Memory T cells induced by previous pathogens can shape the susceptibility to and clinical severity of subsequent infections. All right, let's look at that for just a second. So this is previous pathogens, though. It's not the same pathogen. And so this starts to explain right here, right in the opening sentence. We get this idea around how these memory T cells, the ones that are hanging on to the memories of the past pathogens, that can really shape your susceptibility to these new infections that come along, not necessarily to exactly the same pathogen. And that's the point of this paper, that the coronavirus is Maybe there's some crossover between them in terms of your body seen one coronavirus, which is part of the common cold series, is other ways you could have been exposed to coronaviruses. And maybe it's your body seen that. You happen to have a T cell that recognized a portion of that other coronavirus that's the same as SARS-CoV-2. Bingo, you have higher immunity to this thing. You've got a much better chance of fighting it off more quickly and with fewer effects than somebody who hasn't been exposed. All right, continuing on here. Little is known about the presence of pre-existing memory T cells in humans with the potential to recognize SARS-CoV-2. All right, so they checked it out. Here we first studied T cell responses to structural, so those are proteins, nucleocapsid protein or NP, and non-structural. So these are all these things right here, just so we know, these little things. NP stands for nucleocapsid protein. It's a protein in the outer capsid that surrounds SARS-CoV-2. So the virus is a complex structure. It's made of a ton of different proteins, all of which surround the RNA message in the center of this thing. So this is one of them. The nucleocapsid protein is one of them. And also other proteins like NSP7, NSP13, all very excitingly named, of the open reading frame 1, the ORF1 region of SARS-CoV-2. All right. And so they studied this in what they called Covid-19 convalescents. So that's 36 people who had gotten better from COVID. So they'd been from their COVID disease. They'd been exposed to SARS-CoV-2, got COVID, then they convalesced. In all of them, we demonstrated the presence of CD4 and CD8 T cells recognizing multiple regions of the NP protein. Great. We then showed that SARS recovered patients, this is SARS, classic SARS right here, so let's make sure we don't get confused. SARS here is classic SARS, not SARS-CoV-2. All right. And when they looked at SARS recovered patients, they found that they still possess long-lasting memory T cells reactive to the SARS NP17 protein, the NP protein 17 years after the 2003 outbreak. So these T cells in these SARS, classic SARS recovered patients, were still reactive to the NP protein 17 years after that 2003 outbreak, which is, all right, so that means there's long-lasting immunity potentially. And that's what we're looking for in anything that's going to give us herd immunities. You want long-lasting immunity, of course. And get this, though, that in these SARS, classic SARS recovered patients, their T cells displayed robust cross-reactivity to SARS-CoV-2 NP, the nucleocapsid protein. Surprisingly, we also frequently detected SARS-CoV-2 specific T cells in individuals with no history of SARS, COVID-19, or contact with SARS COVID-19 patients. So looking at so-called normal controls, they found people who had SARS-CoV-2 specific T cells, even though they couldn't find any history of SARS, COVID, or any of this stuff, right? Hey, maybe it's possible these people somehow got exposed to SARS-CoV-2, and we don't know that because maybe this thing just swept through the population way more than we thought. But in fact, they were pulling a lot of these samples, I believe, from blood of people that was preserved. And if I read it right, I think in this case, these are people who may have been sort of what we would call pre-COVID. All right, that's a lot to digest right there. I'm throwing a lot of stuff at you. We got NSP7, NSP13. We got this open reading frame. What are we talking about here? There's a lot of gobbledygook. Let me decode it for you so we don't get lost in here. This is the structure of SARS-CoV-2. It's about a 30 kilobit piece of information. So there are RNA bases. This is a ribonucleic acid virus, all right? And all of those ribonucleic acids are strung along, and we would count each one of them. There's one ribonucleic acid, there's 2, 3, and we get up to around 30,000, and that's the whole thing, all right? And it's arranged like this, where we have this big first polyprotein, which is up to here about 13 kilobases in. We got the second polyprotein. We got the spike protein. That's the part that binds on to the ACE2 and other receptors like the CD147 and looks like neuropilin, things like that. Then there's an envelope protein here, and there's nucleocapsid proteins way out here. So at any rate, this is the whole genome of this little virus that's been giving us so much trouble, and it gets read from one direction to the other. When it's in the cell getting replicated, it gets read. And it's reading as it's being read along, it's being translated. And what is it being translated into? Well, the RNA itself doesn't do anything, but it gets translated and converted, read along like just like you would read a book. Lots of letters strung along, but those letters form words. So the proteins in this story would be the words that we would see. And here's how they're sort of structured along. Again, going from one end to the other, we're reading along this way. We've got the spike protein here. It looks like that as a protein all folded up. There's an endoribonuclease protein here. There's a helicase protein, a replicase protein, a 3CL protease. It has its own protease. It has another papain-like protease. These are all individual proteins, and it's got a bunch of them. All right, and this is, trust me, this is all going somewhere. Gets important and to our understanding of all of this. So over here in open reading frame 1, a lot of these proteins are arranged, and then there's open reading frame 1b or ORF1b. Here's the spike protein, and then all of these other ones. And again, we're almost 30,000 base pairs, 29.9. And all of these individual proteins, like let's look here at the spike protein, this one right here. This is the spike protein. And what happens is the spike protein gets read by the machinery of the cell, converted into a protein, and the protein, here it is, arrayed on the outer surface of the entire virin particle here. And here are other ones. Here's the M protein here. Here's the N protein way in here, things like that. So there are all these different proteins arrayed around, and each one of these proteins is a potential target for your body, your T cells, your B cells to recognize and say, hey, that's not self. That's something else. So let's get rid of that thing. And let's do that. So your body, when it develops immunity to little tiny fragments of these proteins, little tiny chunks, 6, 7, 8 amino acid chunks that it recognizes and says, not self. And then it mounts a vigorous defense against that. All right, let's go into more of the findings from this paper then with that understanding. SARS-CoV-2 T cells in uninfected donors exhibited a different pattern of immunodominance. So immunodominance, what they're talking about here is what is the T cells, what are they looking at? So they have a dominant thing that they're sort of looking at. And some of them will be targeted against this protein and some will be targeted against that protein. But the dominant, what are they really mostly dominantly arrayed against? That's the immunodominance. Frequently targeting the ORF1 coded proteins, NSP7 and 13. So NSP7 and 13 are in the ORF1 region, which means they're getting read first. So as this virin particle comes into a cell, this one right here, and goes and gets dumped into a cell, and it releases, this is its RNA package of information inside, all coiled up in there. As that thing comes out and goes into the cell, it gets read out. And so the proteins here are coming out and being expressed earlier than the proteins down at this end. So these are early ones that are coming out, and these are a bit later. But at any rate, the whole thing is read out. But this is, you're going to get a bunch of these ORF1 proteins first. So this is interesting because in the T cells of uninfected donors, they had a different pattern of immunodominance. And in these uninfected donors, they were frequently targeting somehow already the ORF1 coded proteins NSP7 and 13. As well as the NP structural protein. Epitope characterization of NSP7 specific T cells showed recognition of protein fragments with low homology or similarity to common cold human coronaviruses, but conserved amongst animal beta coronaviruses. And of course, COVID is caused by SARS-CoV-2, which is a beta coronavirus. Thus, infection with beta coronaviruses induces multi-specific and long-lasting T-cell immunity to the structural protein NP. Let me decode that for you really quickly. So if you've been exposed to a beta coronavirus and you mount a defense against this NP protein here, this multi-specific part means that any other beta coronavirus that comes along, you're going to have a defense against it because your T cells have already seen this same structural protein NP, and that's because it's highly conserved. That means it's not different. It's not like SARS-CoV-2 has one version of it, and MERS has a different version, and other beta coronaviruses have a different version. This is a really important protein. You monkey with it, and the virus is no longer viable, so they conserve it. It's a very highly conserved region of that particular protein. Understanding how pre-existing NP and ORF1 specific T cells present in the general population impacts susceptibility and pathogenesis of SARS-CoV-2 infection is of paramount importance for the management of the current COVID-19 pandemic. So yes, how is it that some people have this pre-existing specific T cells? Where did they come from? How can we induce more of those? And then secondarily, we'd like to know, and we don't have this data yet. This paper didn't go that far, where we could actually see, and this would be great to find out, if they could pull out the T cells from people who had known clinical experiences like mild, severe, dead, and check their T cells and see how they fell out. And if there's a clinical correlation, meaning people whose T cells have a very high pre-existing NP and ORF1 specificity, well then great. If those people also have very mild clinical cases, now we have an angle that we can go down and say, okay, how would we get more of these T cell responses? You know what? It might be that we expose people to other beta coronaviruses that aren't SARS-CoV-2, that don't cause COVID or any other significant disease. Maybe, who knows? It might be that people want to get the common cold and get a good old case of it because your T cells are all geared up against those coronaviruses if that turns out to make you more resistant to SARS-CoV-2. All right, continuing on. Severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, is the cause of the coronavirus disease. This disease has spread pandemically, placing lives and economies around the world under severe stress. SARS-CoV-2 infection is characterized by broad spectrum of clinical syndromes, ranging from asymptomatic or mild influenza-like symptoms to severe pneumonia and acute respiratory distress syndrome. It is common to observe the ability of a single virus to cause widely differing pathological manifestations in humans. Let's read that again. It is common to observe the ability of a single virus to cause widely differing pathological manifestations in humans. This is often due to multiple contributory factors, including the size of the viral inoculum. Hold on, we're going to get back to that in just a second because you're absolutely right about that. Size of the viral inoculum. Hold on to that thought for a second. The genetic background of, the genetic background of the patients and the presence of concomitant pathological conditions. Those would be our comorbidities. That's concomitant pathological conditions. Comorbidities. Moreover, an established adaptive immunity towards closely related viruses or other microbes can reduce susceptibility or enhance disease severity. And by the way, adaptive immunity towards other related viruses or other microbes. We've heard things about the BCG vaccine, which is a vaccine for tuberculosis. It seems to be that people who have been so challenged with that BCG vaccine are more resistant to COVID, and possibly because of this thing that they're talking about right here, which says the established adaptive immunity to other microbes can reduce susceptibility or enhance disease severity. So interesting stuff going on here. Wow, is this ever complicated how the immune system works? So that's why we say make sure your terrain is as robust as possible, especially the vitamin D. Make sure you got a lot of vitamin D. All right, remember this thing, the size of the viral inoculum right here. Well, that's why we keep saying, how do we get back to our lives? We wear masks. It's not hard. Everybody wears a mask. Because if you have a mask, you are no longer a spreader. You don't wear a mask to prevent you from taking in what other people have coughed out so much as to prevent you from coughing out your particles into the world. It helps us cover our face holes because that's right. Remember, this virus doesn't come in through our face. It comes in through our mouth, our nose, our eyes, right? So a mask at least covers the most important holes that we've got there. But the big thing here too is that we've always said you have better odds of a lesser illness. Why? Because you get that low inoculum, all right? You get a smaller, if you're going to get this thing, if it happens anyway, you want to start with a low load, a low dose of this thing, not a high dose, because we know that contributory factors includes the size of the viral inoculum. We've known that for a long time. All right, carrying on here. SARS-CoV-2 belongs to the Coronaviridae family of large RNA viruses infecting many animal species. 6 other coronaviruses are known to infect humans. 4 of them are endemically transmitted. Endemic means they're just resident in our population. We catch them all the time. They're around. And cause the common cold. And those would be the coronaviruses OC43, HKU1, 223E, and NL63. Very super exciting names. Scientists rock at the naming thing. I mean, you got to love those, right? All right. While SARS-CoV, defined now as SARS-CoV-1, that's classic SARS, and MERS-CoV have caused limited epidemics of severe pneumonia, all of them trigger antibody and T-cell responses in infected patients. However, antibody levels appear to wane faster than T-cells. So again, the vaccine with making antibodies might not last. That's the concern. We don't know yet. Maybe these vaccines will be amazing. Maybe we just get a booster every year and we're fine. We don't know. But that's the concern. SARS-CoV specific antibodies dropped below the detection limit within 2 to 3 years. So that's SARS classic. So again, the antibody doesn't seem to last for this particular coronaviruses. Whereas SARS-CoV specific memory T cells have been detected 11 years after SARS infection. That's all really cool. Since the sequences of selected structural and non-structural proteins are highly conserved amongst different coronaviruses, especially for example, NSP7 and 13 are 100% and 99% identical, respectively, between SARS-CoV-2, SARS-CoV-1, and the bat SL-CoV VZXC21, which is very distantly related, except that these proteins right here, NSP7 and 13, are, look at that, 100% and 99% identical between all of these things. All right, we investigated whether cross-reactive SARS-CoV-2 specific T cells are present in individuals who resolved SARS-CoV-1, comparing responses with those present in resolvers of SARS-CoV-2 infection. We also studied these T cells in individuals with no history of SARS or COVID-19 or of contact with SARS-CoV-2 infected cases. Collectively, these individuals are hereon referred to as SARS-CoV-1-2 unexposed. All right, so there's a lot of stuff to go through, but I'm really interested in this idea that these proteins up here are really highly conserved, and that when the body's mounting a long response to something, it's very wise, and I don't know how nature is this wise, but it chooses to mount a response against these particular proteins. And let's check this out. So here we're looking at these letters here are all amino acids, and we have here seven different things. We're comparing SARS-CoV-2, which is the causes COVID-19, which we're facing now, classic SARS, MERS, and then also these last 4 things right here. These are all the common cold viruses. And so we're comparing the genetic structures as expressed in their amino acids across these different things. So this is the NP protein. And you can see here, all the way down in this region, look how highly conserved this is. It's very, very, very identical down here. So if your body mounted a response, particularly to this little string right here of these 7 amino acids, you would have the same robust response, regardless of whether you'd been exposed to the common cold, or SARS, or SARS-CoV-2, or any of that stuff. Looking at the same sort of presentation for NSP7, you can see that fairly highly conserved regions like this one here, get this right pen. This is very highly conserved region down here. There's other really pretty strongly conserved regions we can look at here. And of course, SARS-CoV-2 and SARS-CoV-1, very, 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 very similar. So it's no surprise that SARS-CoV-1 patients have a very strong response to SARS-CoV-2 because their T cells at least, because they would have seen something very, very close to that. And as well, let's look at this one. This is NSP13, and again, super highly conserved. Look at all the yellow matching in here. That's just fantastic. Really highly conserved down here. So it's pretty interesting that, yes, you would want to look at these areas, and if somebody's been exposed to the common cold like this, and they mounted a response, their T cells mounted a response to NSP13, you would have a very strong chance, the theory would go, of then recognizing SARS-CoV-2 and mounting a better, more complete, more elaborate response. So that's the theory. That's what these people are looking at here. And so they a lot of data in this paper, really good. I don't have time to go through it all, but I thought that's a really fascinating idea that we're going to track and we want to look at more. It's very hopeful, by the way, because now at least we have something we can begin to pinpoint and start testing clinical experiences against actual substances, in this case T cells, from patients' bodies. Conclusions were really interesting here. It's why NSP7 and 13 specific T cells are detected and often dominant in SARS-CoV-1-2 unexposed donors, while representing a minor population in SARS-CoV-1-2 recovered individuals is unclear. So they found one difference there, which was, hey, they found these reactive cells, but in a dominant form in unexposed donors, but they didn't really see that in the SARS-CoV-1-2 recovered people. So that was a little bit of that was a little odd to them. It is, however, consistent with the findings of Grafoni et al., who detected ORF1 specific T cells, preferentially in some SARS-CoV-2 unexposed donors, while T cells of COVID-19 recovered patients preferentially recognized structural proteins. This is starting to hone in a little bit because these ORF1 reading area, remember I told you that ORF1 is that first region, it's that first region that gets read in here. And because it's that first region that's read in here, it means that these are some of the earliest proteins to get kicked out or made by the virus. And by the way, that would be great. You want your body to recognize as soon as possible that a cell has been infected with this virus. So targeting those ORF1 frames would make a lot of sense. All right. Induction of virus-specific T cells in exposed but uninfected individuals has been demonstrated in other viral infections. All right, that's pretty cool. Induction of virus-specific T cells, exposed but uninfected. Okay, we've seen this in other viral infections, so we have a theory working on here. Theoretically, individuals exposed to coronaviruses might just prime ORF1 specific T cells, since the ORF1-coded proteins are produced first in coronavirus-infected cells and are necessary for the formation of the viral replicase transcriptase complex essential for the subsequent transcription of the rest of the viral genome, leading to various RNA species. All right, so you need the ORF1 products first. It makes a lot of sense that the virus would put those up front because you need those products first to make the stuff that comes later. So it makes a ton of sense. Therefore, ORF1 specific T cells could hypothetically abort viral production by lysing, destroying, smashing open SARS-CoV-2 infected cells before the formation of mature virions. In contrast, in COVID-19 and SARS patients, the NP protein, which is abundantly produced in cells secreting mature virions, would be expected to result in preferential boosting of NP-specific T cells. So, all very complicated, but here's the punchline. It turns out that your body is very, very quickly figuring out that you've got an infection, and the way it goes about clearing that out, one of the ways is that it's looking for these things that are here made in the ORF1 open reading frame. Here's where NSP7 is made. It's right solid in this green zone. NSP13 is right here. This is just an 83 amino acid protein, not that huge. NSP13, 601, that's pretty getting big. And of course you could mount antibody responses against, the spike protein or all these things that are on the outside, but these early ones are produced in the cell and they get presented to the outside. The cell will package them up and put them on the outside and go, hello, help here. And your T cells will come along and see those on the outside of the cell and go, uh-oh, this something's wrong here. And they will kill that cell, and or target it for elimination. So pretty cool. Importantly, the ORF1 region contains domains that are extremely conserved among many different coronaviruses. That implies that if you've had the common cold, it might be that you are going to be more resistant to this particular disease when it comes along. The distribution of these viruses in different animal species might result in periodic human contact, inducing ORF1 specific T cells. With cross-reactive abilities, in this case against SARS-CoV-2, understanding the distribution, frequency, and protective capacity of pre-existing structural or non-structural SARS-CoV-2 cross-reactive T cells could be of great importance to explain some of the differences in infection rates or pathology observed during this pandemic. T cells specific for viral proteins have protective ability in animal models of airway infections, but the impact that pre-existing NP or ORF1-specific T cells could have in the differential modulation of SARS-CoV-2 infection will have to be carefully evaluated. So, we don't know yet, but it's pretty fascinating that they have managed to find that there are people out there who have T cells that are capable of recognizing SARS-CoV-2 even though they have never seen it. Also interesting that people who had SARS 17 years ago still have T cells that are like, yep, I recognize that. So two things we want to see. It hints at a path for us to get towards actual immunity to SARS-CoV-2 without catching SARS-CoV-2. Maybe we could be exposed to a different virus, and that would give us the same immunity. That would be fantastic. Give me a common cold, and I can avoid getting SARS-CoV-2. I'm in, right? So that's awesome. And the second thing is, it points to the idea that this is long-lasting potentially, so that we have SARS-CoV-2 recovered, classic SARS recovered patients with this immunity apparently in their T cells. At least they can still recognize this thing 17 years later. Obviously, we haven't exposed these people to SARS, classic SARS, to see if they get it again. But the idea here is that when you have that portion of your immune system, the adaptive immune system that is sitting there with long low memory, waiting to see this thing again, and that can confer a lasting immunity, that's fantastic. So the T cells might be a path that we really need to understand a little bit better, but it's great to see this research coming along. And it really starts to, as soon as I read it, it started to snap into place, because it's really been very confusing. Why is it that so many people get this really horrible expression of this thing, and why do some people not get it nearly as much? Why are children seemingly immune to this thing relative to older people? Well, the T cells begin to actually help us understand that, because the T cell responses are something that you would imagine would be more robust in a younger person than an older person. And so anyway, lots to unpack and talk about over time, but this is something I'm going to be tracking as time goes on. Last time, which was on the 14th, I talked about this one, scans reveal heart damage. A number of you pointed out how I misread this table in terms of what I said. I'm sure I did. This 15% number in particular right here, that was 15% of the overall number, not 15% of people with mild symptoms. So let's get the numbers right, and thank you for everybody who sent this in kindly. For those of you who sent in an unkind, you got that wrong a bit. I ignored you, so you didn't get a response from me. And for people like Steve, who sent in a really nice response, thank you very much. And here's the corrected numbers, because I like having corrected numbers. So of people who had mild symptoms, mild COVID symptoms, turns out 45.6% of them had an abnormal heart scan. And the numbers break out like this. With moderate, people had 64.2% showed up with a abnormal scan. And in the severe COVID cases, it drops back down again to 54.4% had an abnormal heart scan. Don't know why that is. You would expect these numbers to kind of go up as you went this way. The fact that they don't have an explanation for that yet. Possible that a lot of the people who were in the severe category didn't survive, and maybe they died from heart issues. So that's a possibility. So maybe there were just fewer people surviving with serious heart conditions or abnormalities down here. Who knows? I don't know that yet. The other thing I can't quite find, though, is the data that would say how many people of this age with these comorbidities would have also abnormal scans. Don't know that answer yet. So I think it's going to be a reasonable number. I found one study that said it was around 20 to 30% of people were showing up, but I couldn't get ages off of that, and they weren't matched for comorbidities. So I don't know. It's just, are these numbers? So here's what I'm saying: are these numbers really too high, or on average, does about half the people who are of this age with these comorbidities, would they also have what would be considered an abnormal heart scan? Don't know. But I read into this paper much more completely. Couldn't tease out the data I wanted. So we're going to just keep watching that. Thanks again for everybody who sent in. Noticed that error, and I'm glad to correct it here. So remember, asymptomatic does not mean that everything is okay. We know that there's fairly long lasting. We don't know if it's permanent damage, but the lungs, certainly if they're fibrotic, we don't know of any cases from other diseases where fibrosis of the lungs reverses. It's scar tissue in the lungs. The lungs are incapable of replacing scar tissue. Heck, your skin, which is a genius at replacing itself, often has scars that last for a lifetime if they're bad enough, deep enough, all of that. So at any rate, we see that long-lasting damage. And again, that's why I don't want to get this. Not until we understand more, not until we have something that approximates a reasonable treatment cure that I can believe in. All right, conclusions and summary for today. Vaccines, hey, those give us antibodies. But it's really unclear still how, when, or if those are going to provide any lasting protection. The data from other coronaviruses, including classic SARS, says the antibodies don't last. So, hey, maybe not that bad a deal if the vaccines just, you have to get a booster every year or two or something like that. Maybe that's how we go through this. I don't know. But for now, I'm still kind of skeptical about the whole vaccine thing. I'm watching it. I'm not super rah-rah, yay, those are going to come along and save us all soon. Maybe, but I got to be honest. I'm going to watch. They're being rushed through big time. They're being rushed through production, and I'm very skeptical of cutting corners when it comes to messing with the immune system. Personal opinion there. All right, T cells, on the other hand, they seem to provide long-lasting memory, presumably protection against SARS and MERS, as well as other beta coronaviruses. And so that leads to the conclusion that people's different T cell histories may explain the continuum of COVID-19 severity. And it might be something as simple as your body was exposed to a cold, your body happened to target one of these highly conserved regions on one of these other viruses. And therefore, when you saw SARS, your body's like, seen that, know what to do. And other people had different genetic draws and were less lucky, and maybe their body mounted an attack against a very different region on a cold virus if they'd been exposed at all, and therefore they didn't have any competence against SARS-2 when it came along. Their body was naive and didn't know how to process it, and came up with a bad case of COVID. All right, which means lots more work to unravel all this, and that typical vaccines may not work at all that may not work all that well, or maybe they will. I don't know. We're just gonna have to find out. Remember, I didn't go through all of the horrifying data come out of the United States with climbing numbers of cases, deaths, all that stuff in a couple of key sentinel states, Arizona, Florida, etc. But it didn't have to be this way. So many other countries have this under control. So many. And so we have the blueprint for this. We know how to do it. Hey, you lock people down and you pay them to stay home. And guess what? Everybody wears a mask. You get through this. The United States looked at all of that and said, we don't understand what you mean by any of that. So we're just going to keep doing what we're doing. And it really doesn't have to be this way. So truth on this billboard up here. I hope they keep that billboard up for a long time. Remember, if you get a t-shirt, you can join a movement, and we have all these wonderful pictures flowing in of all these different people wearing their resilience t-shirts. We want you to be happy, healthy, whole, resilient. You can still throw a few things in the ground here in the middle of July and get food by fall. Remember, one of the most important things you can do is to make sure that your immune system, the terrain of your body, is as tuned up as it can be, because, wow, it's wildly complicated. A lot of moving pieces in there. T cells, B cells, dendritic cells, macrophages, all that stuff. Tons of things, and they need various macro and micronutrients to function, and vitamin D3 might be the most important of all of those, getting vitamin D in your body. All right, that's all I have for you today. Thank you so much for listening. I know that got a little sciencey, but the science is where the fascinating part of this comes in. And it's just fascinating to watch this get unraveled. And we're going to learn a lot. And by the way, this is going to help us be healthier in the future against all sorts of other viruses, because we're learning so much so rapidly at this time in history. So that's the good news about all this. I think we're going to conquer lots of other viruses as a consequence of this. But unraveling the T cells is going to be a critical component of that. Lots more attention is going to be coming to that. That's a good thing. That's how this is supposed to go, and I'm glad to see that work being done. Again, thanks for listening. We'll be back with you next week. Bye-bye.
Hi, folks. This is Adam Taggart, Chris's co-founder at Peak Prosperity. To get your own Resilient shirt, simply go to peakprosperity.com/shirts. Each shirt is made of 100% organic cotton and produced in the USA. And when your shirt arrives, Please send us a photo of you wearing it if you feel comfortable doing so. We'll add you to our wall of proud Peak Prosperity members who are showing the world that resilience is the way to a better future. Thanks for watching.